to continue with our discussions about the cell organelles, we go now to Golgi Apparatus. Golgi Apparatus, that is a network of flattened, smooth membranes and vesicles. So that is a converging area of endoplasmic reticulum, wherein Golgi Apparatus is located near or beside only the endoplasmic reticulum. It is composed of cis, medial, trans cisternae. Main function: protein sorting or arrangement, packaging, secretion, maturation of synthesis of proteins. So if this is the endoplasmic reticulum, proteins are released and enters the cisternae of Golgi Apparatus. So protein progress through Golgi stack in cis-trans direction. So each stack of flattened Golgi sacs has several defined regions. The cisterna nearest the ER, cis, the centrally located medial, the trans, or the Golgi Apparatus, trans Golgi Apparatus network at the end portion. And this one is the proteins that is being released when proteins are matured already in Golgi Apparatus. So try to look at our video diagram presentation. So we have endoplasmic reticulum releases the proteins that is transported by a vesicle to the cis Golgi Apparatus, then passes to the medial, then trans, and then it is the vesicle will going to receive the proteins and supply to the plasma membrane, because the plasma membrane needs more proteins for the structural framework, of the plasma membrane. It needs more proteins, and some proteins are also digested by the lysosomes. Next, we go now to lysosomes, tiny organelles that is found in the cell cytoplasm. Okay, cell scavenger are known as cell incinerator because they digest foreign chemicals or food, and even the damaged organelles, composed of bag of enzymes, and it will going to degrade proteins, carbohydrates, lipids, and nucleotides, or known as the biomolecules. So these are the two functions of lysosomes, the phagocytosis and autophagy. Plasma membrane forms the invagination known as food vacuoles that will going to receive the food or other chemicals that enters the cytoplasm of the cells, and then the food vacuoles carries the food, and then it will diffuse with the lysosomes, because lysosomes here contains bag of enzymes. Then food vacuole fuses with lysosomes, and hydrolytic enzymes digest the food particles. So digestion process takes place in the fusion of lysosomes with the food vacuoles, and then the enzymes will going to digest the food. Okay. For autophagy, so vesicle containing damaged mitochondrion, for example, or mitochondria, lysosomes fuses with the vesicles containing the damaged organelles, and then hydrolytic enzymes will going to digest the organelles. So this is the two main functions of the lysosomes. So lysosomal enzymes have an optimum pH of 5, so that is almost acid or weak acid. Okay. Polysaccharide hydrolyzing enzymes, we have alpha glucosidase that will hydrolyze glucose. We have alpha fucosidase or fucosidase will digest fucose, and beta galactosidase digest galactose. Alpha mannose, it will digest or hydrolyze mannose. Hyaluronidase will digest hyaluronic acid. So aryl sulfatase enzyme involved in sulfation process, and lysozymes involved also as antimicrobial property. Okay, usually it digests also foreign chemicals. Furan hydrolyzing enzyme, protein hydrolyzing enzyme via catepsins that will digest proteins. Collagenase digest collagen. Elastase will digest elastin, the yellow fibers. Peptidase will digest peptide. So peptide is amino acid plus amino acid. So the enzymes that will going to digest or hydrolyze is the peptidase enzyme. We have nucleic acid hydrolyzing enzymes, ribonuclease and deoxyribonuclease. So this one ribonuclease will digest RNA or ribonucleic acid. Deoxyribonuclease will digest deoxyribonucleic acid or DNA. We have lipid hydrolyzing enzyme. So we have fatty acid esterase enzyme. It will hydrolyze also fatty acids. Phospholipases. Okay, these are enzymes that involved in phosphorylation process. Next, we have peroxisomes, granular matrix. So the size is 0.3 to 1.5 micrometer. So it contains peroxidase enzyme. Okay, and it contains also catalase enzyme. So these are enzymes responsible for the oxidation process. Prominent in leukocytes and platelets. Usually peroxidase and catalase usually involved in the oxidation of or destroys the unwanted peroxides in other radicals. So what are the other radicals that is found in the body? Usually the other radicals found in the body is yung mga acids, aldehydes, ketones, okay, alcohols, and others. So they are known to be radicals. Radicals are also the product of metabolic property. Okay. So these are the anatomy of your peroxisomes. It has also plasma membrane, lipid bilayer phospholipids, ba? Urate oxidase, crystalline core, okay, for oxidation process. Next, mitochondria, spherical oval rod-like shape. So take note, the diameter is 0.5 to 1 micrometer. So up to 7 micrometers. So erythrocyte does not contain mitochondria. So the tail of spermatozoa is fully packed with mitochondria. Okay? The tail kasi ng spermatozoa or sperm cell, usually it is the used for swimming purposes, so it needs more energy. That's why mitochondria is occupies the tail portion of the spermatozoa or sperm cell. I mean, powerhouse of the cells, because energy is being utilized and produced in the form of ATP. Okay, so this is the mitochondria. It has inner membrane, the outer membrane. Contains also DNA, granules, ribosomes, cristae, inner membrane space, matrix, and ATP synthase particles, which utilize the generation of energy in the form of ATP. So cytochrome P450 system, so mitochondria is inner membrane is involved in steroidogenesis on the production of steroids. If you try to see the tail portion of this sperm cell or spermatozoa, it is composed of mitochondria because it needs energy for swimming and movement purposes. Okay, so superoxide dismutase is present in cytosol in mitochondria. So this one is also responsible for oxidation process. Antibiotic inhibiting bacterial protein synthesis does not affect cellular processes, but do inhibit mitochondrial protein synthesis. Take note, it will cause damage to the bacterial cells, because in the principle involved wherein antibiotic will only inhibit the multiplication process, is just inhibiting the protein synthesis inside the cells. But inhibiting the mitochondrial protein synthesis, it will cause danger to bacterial cells because there is no energy generations that will affect the functions of bacterial cells. So that is very important based on the spectrum of actions of antibiotic. So protein synthesis there, that is already biochemistry aspects. Okay? Next, plasma membrane. So it separates the cell from external environment. Okay, if this is the cell, so this is the internal part, intracellular. We have the extracellular part. So this is the bilayer phospholipids, semi-permeable because most water or fluid permeates to this bilayer phospholipids, and it is metabolically active, very active, because of the different materials that crosses the cell membrane. So try to see, the cell membrane is composed of different protein. We have glycoprotein that is responsible for the transport of your glucose, integral protein for communication process, transmembrane protein for communication and entry of different molecules or ion, and this is also known to be the ion channel proteins. We have the hydrophilic phase or the water region phase, and the hydrophobic phase, or known to be the lipid phase. So one characteristic of plasma membrane is known to be amphoteric because it possesses both hydrophilic region and hydrophobic region. Based on the fluid mosaic membrane model, this model refers to that the cell membrane is floating like a boat. Okay, that is a fluid mosaic theory. Ectoenzymes, nucleotide phosphatase and alkaline phosphatase enzyme are also located in that membrane. That is, they involved in phosphorylation process, made up of lipids and small amount of carbohydrates. Next, your transport mechanism. So transport mechanism, we have passive transport, simple diffusion, facilitated diffusion, ion channels, active transport, and different pumps. Okay? For simple diffusion, we have simple diffusion, movement of molecules from higher to lower concentration across the cell membrane. So this is a transport mechanism that follows the law of mass actions. It does not require energy. So the rate of entry is proportional to the solubility of solute in the hydrophobic region. So meaning the size of the smaller the size of the particles that crosses the bilayer of phospholipids are faster compared with larger size or molecular weight. The lower the molecular weight, the faster is the diffusion process. So that is follows the law of mass action. Next, facilitated diffusion. So facilitated diffusion, it is carrier mediated. It is the movement of particles from higher to lower concentration. Okay, that is facilitated by a carrier known as a protein. Carrier exists between two conformations. Okay, there is opening and closing of the carrier. Okay, once the biomolecules, for example glucose, the carrier is glucose transporter. This is the protein. All transporter are proteins. So there exist two conformations: the opening of that carrier and then the closing of the carrier. Once the biomolecules or other compounds pass through that carrier or being carried by the transporter from outside environment of the cell down to the inside environment of the cell. So more rapid compared to simple diffusion. Yes, once the example, the biomolecule fits to that carrier, so it transport quickly. It has a ping pong state. So the once the biomolecules carried and received by the transporter, then the exit or the efflux is considered to the Pong states. So glucose transport across membrane is facilitated by glucose transporters known as proteins. Next, we have ion channels. So these are known to be transmembrane protein. So meaning transmembrane protein, they are found across the cell membrane or the surface of cell membrane. See, this is the bilayer of phospholipids. Okay? And selective to one particular ion, meaning kung sodium ion siya or calcium ion or chloride ion lang siya. So that is known to be selective. So regulation activity done by, there is voltage gated, ligand gated, mechanically gated. So we will discuss one by one. So the transport is also very quick. It's almost the same as facilitated diffusion. So it does not require an energy. So once particles or molecules or ions, reaches the channel and it is very specific. Example, sodium. So the sodium will open and then ions will enter. So that is specific only for sodium ion, for example, or calcium ion. Next, active transport. So this one, movement of particles across the cell membrane from lower to higher concentration, against the concentration gradient. So, pwede magtawag natin na one direction lang yon, or unidirectional, meaning one direction. So, transport requires energy. 40% in the cell are used. So, it requires energy in the form of ATP. So, ATP is converted to ADP plus inorganic sulfate, meaning one phosphate is being utilized from ATP. So, during the transport of the ions, so there is utilization first of energy, or energy is being used in order for the transport to proceed. Okay? So, cardiotonic drugs like digoxin and ouabain are transported by this mechanism known as active transport. So,